All right, I think I'm gonna get started. I'm gonna introduce myself because I think Jeff is in the other building. My name is, oh, he just texted me. Yep. Um, yeah, I'll ask him if he can text. It's probably on the other side. Um, so my name is Martin Felices. I'm an assistant professor in the Division of Hematology, Oncology, and Transplantation. Been an assistant professor for, I think, 2.5 years now. I'm also the co-director of the Translational Therapy Lab. I came from UMass. That's where I did my graduate research, and then I went to industry because I wasn't sure what I wanted to do for a while, and I worked for 3 years in Novartis Pharmaceuticals in Cambridge. Then I came to do a postdoctoral fellowship with Jeff, and 3 years later, I joined the department. My interest is in natural killer cells and basically trying to drive them to kill tumor targets better. And today I'm going to talk to you about a story that I think it's pretty complete in terms of driving a molecule from basic research almost all the way to the clinic. We're almost there, not quite there though. So it's not those kind of TriKEs. And I should say it took me an embarrassingly long amount of time to make this drawing. So I'm glad that I'm not a graphic designer. But TriKEs stand for trispecific killer engagers, and they're molecules that engage activating receptors on natural killer cells and engage tumor antigen on tumor cells. And they also have a cytokine to drive better NK cell functionality. So I wanted to talk about a little background on NK cells first. NK cells are involved in reproduction, autoimmunity, viral infection. They also form a bridge between the innate and adaptive immune responses. We utilize them quite a bit in the hematopoietic stem cell transplantation system as a form of therapy, particularly for heme malignancies. But today, we're going to focus primarily on their role in tumor surveillance and cancer clearance. They represent about 5 to 15% of circulating lymphocytes, and I should point out that today I'm focusing primarily on human natural killer cells. They're not MHC restricted. They don't require priming, and they contain preformed granules. They have a broad repertoire of activated and inhibitory receptors, but they don't have any clonotypic receptors like T cells. They mediate their functions very quickly through lytic enzymes, particularly granzymes and perforin. They can also mediate their function through death receptor ligands, cytokine secretion, and antibody-dependent cell-mediated cytotoxicity, which we will talk about quite a bit today. So like I mentioned, they have a lot of activating receptors on their surface. They have a lot of inhibitory receptors, chemotactic receptors, cytokine receptors, and adhesion receptors. And today we're going to be focusing particularly on CD16, which is the NK cell receptor that mediates antibody-dependent cell-mediated cytotoxicity. CD16 engages the Fc portion of antibodies bound to tumor cells or to viral infected cells, and it drives NK cell activation. And it's a very potent receptor, and amongst NK cell activating receptors, it's probably one of the only ones that can drive NK cell activation all on its own. And basically, the way NK cells decide whether or not they're going to kill a target is via balance between activating receptors, here in green, and inhibitory receptors, here in red. And I'll talk about it a little bit more. We're also going to talk about quite a bit about IL-15 signaling today, and I just wanted to let you know that NK cells do have the components necessary for IL-15 signaling. So how do NK cells make a decision whether to kill a target or not? So basically, under normal autologous situations, an NK cell will encounter activating ligands in the surface of targets, but it will also encounter inhibitory ligands, particularly in the form of self-MHC. So NK cells in mice, they have LY49 receptors. In humans, they have Q receptors that engage self-MHC, and that inhibitory signal basically tells the NK cell not to kill. And again, it depends on the balance between activating signals and inhibitory signals. So you can flip that balance by generating more activating signals or by generating more inhibitory signals. Upon infection or transformation, a lot of target cells usually decrease the amount of self-MHC that they have in their surface, and that allows for the balance to tip over to activation and away from inhibition, causing lysing of the tumor or virally infected target. And we use a lot in the clinic allogeneic settings in which we have mismatched donors being transplanted into patients. And what this creates is basically a mismatch in the self-MHC and the inhibitory receptor, allowing to tip the balance against our activating signals. So Melissa Geller, who's one of my collaborators, presented a very similar slide last week. But what I wanted to point out is that as NK cells develop, they go from a highly proliferative potential to highly cytotoxic potential, losing proliferation. And we can see this from high expression of CD56 on NK cells to lower expression of CD56, and basically downregulation of NKG2A, CD94, and upregulation of KIR, but more importantly, CD16. And the most mature NK cells, which express CD57, have high levels of CD16 in their surface. So what are TriKEs and why is CD16 important? TriKEs are composed of two single-chain variable fragments, one coming from an anti-CD16 antibody, which is geared to activate NK cells, and one coming from a tumor antigen. And in this case, we're looking primarily today, I'm going to talk about CD33 and myeloid malignancies. So our previous platform consisted of a single-chain variable fragment against CD16 and one against CD33, bound together by an HMA linker. And this is a flexible linker, and what it allows you to do is it allows to generate a synapse between the natural killer cell and the myeloid tumor cell, so an AML cell, MDS, systemic mastocytosis cell. But we changed this platform by adding an IL-15 linker in the middle, and this is a biologically functional IL-15 linker. So we still have the anti-CD16 to activate the NK cell. We still have the anti-CD33 to target the AML target. But now we have IL-15, which is able to drive proliferation, priming, and survival of natural killer cells. And as you can see over here, we don't know exactly how the IL-15 is functioning. Is it being trans-presented? Is it being cis-presented? Or are there some times where IL-15 is engaging and CD33 is not engaging? We're not sure of that, but as I'll show you, it works. And basically, what the anti-CD16 does in this molecule is it tips the balance towards activation. So we think we can bypass the inhibitory signals by adding a very strong anti-CD16 signal here. And again, CD16 mediates the activation, IL-15 mediates proliferation, survival, and priming, and CD33 we're using it to target AML, acute myeloid leukemia, myelodysplastic syndromes, MDS, and systemic mastocytosis, which is a more rare disease, but we're starting to study that now. Oops. Uh-oh. I think it got stuck. Oh, no. That's not good. Yeah. Okay, here we go. There we go. Okay. So the way we test these molecules is by doing a chromium release assay. And in a chromium release assay, what we do is we load tumor targets with radioactive chromium, and we incubate them with natural killer cells alone or natural killer cells plus a variety of different molecules. And in this assay, we're doing, oh boy, my pointer is not working great. Okay, I'll switch over to this pointer. In this assay, we're doing no molecule, an anti-CD16 scFv, so this is not a full antibody, an anti-CD33 scFv, the BIKE, so the bispecific killer engager with 1633, or the TriKE, which is the trispecific engager with anti-CD16, IL-15, and CD33. And what we see is that the TriKE mediates much more tumor killing, and these are HL60 targets are an AML cell line. So it mediates much more tumor killing than the BIKE, which mediates more tumor killing than all of the controls. So to test, okay, this is working now. To test specificity, we decided to evaluate the tumor killing in a CD33 negative colorectal cancer cell line, which are HD29 cells. And the TriKE is over here. So as you can see, if CD33 is not expressed on the tumor cell line, we don't get killing. But to verify that we could kill this cell line, we generated an EpCAM TriKE that contains an anti-EpCAM single-chain variable fragment. And EpCAM is highly expressed in this line. And as you can see, the EpCAM TriKE did mediate tumor killing very efficiently. So this tells us two things, right? That the molecules are specific, and it also tells us that we can use this as a platform. We can flip out the tumor antigen, put in a different tumor antigen, and it will be effective in killing of the cell line that contains that tumor antigen. Next, we evaluated the ability of the TriKE to induce cytokine secretion. And as you can see here, and this is an ELISA assay, basically we're just looking at the supernatant. As you can see, the TriKE induced much better interferon gamma, TNF alpha, GM-CSF, and MIT1 alpha than the BIKE or no treatment. So this indicates that it can also drive inflammatory signals quite well. This is working really slow. There we go. Next, we evaluated the ability of the IL-15 within the TriKE molecule to induce survival of natural killer cells. And what we did is we took natural killer cells, we put them in culture for 1 week with either the BIKE molecule, the 1633 BIKE molecule, or the TriKE molecule. And what we did at the end is we used a dye that incorporates in dead cells, basically live dead. I think a lot of you use this. And what we saw is that the BIKE molecule basically did not protect the NK cells at all, and most of the cells died in this assay. But the TriKE induced survival in most of these cells, and this can be seen by lack of incorporation of the dye. Next, we wanted to see if the IL-15 moiety on the TriKE molecule could drive proliferation of NK cells, and if it was specific. And what we did is we cell trace labeled peripheral blood mononuclear cells, PBMCs. Cell trace is very similar to CFSC. It's a dye that gets incorporated very quickly, but it divides in half with every division. And we waited for a week, and again, we treated these with TriKE molecule, and then we evaluated proliferation on T cells, which is this line over here. We did not see very much proliferation on the T cells, and on NK cells, and as you can see on the NK cells, it drove a lot of proliferation. Now, I should say, if we do this with IL-15 alone, we see proliferation on the T cells. So what we think is happening here is the anti-CD16 is targeting the IL-15 to the NK cells. And this is important in terms of specificity because it allows you to more specifically deliver the IL-15 to NK cells. So next, we wanted to evaluate if there's any synergy between the CD16 signal and the IL-15 signal in the TriKE molecule. And what we did, and actually DFAF in the lab did this, we evaluated STAT5 signal and phospho-STAT5 signaling by flow cytometry. And we did the assay in two different ways. One, we just added the NK cells with a variety of treatments. And the second way, we added the NK cells plus tumor targets plus a variety of treatments. And when we add the NK cells alone, medium doesn't induce STAT5 signaling. IL-15, as expected, does induce STAT5 signaling. The BIKE doesn't induce very much of it. BIKE plus IL-15, you get very similar STAT5 signaling to IL-15 alone. and the 16-15-33 TriKE induces the same amount of STAT5 signaling when tumor targets are not present. But when we add tumor targets, we see a difference. So NK cells plus tumor targets alone, we don't get very much STAT5 signaling. IL-15 gives us about the same amount of STAT5 signaling as we see without tumor targets. The BiKE plus tumor targets, somewhat surprisingly, induces STAT5 signaling to a certain extent. But the interesting thing is when you do BiKE plus tumor targets plus IL-15 or TriKE plus tumor targets, we get an enhanced induction of STAT5 signaling, indicating that there's some synergy between the CD16 signals and the IL-15 in the TriKE or when you add it separately with a BiKE. There we go. So the next step was to evaluate if these molecules work in post-transplant patient samples. And we look at post-transplant because we use it therapeutically in AML systems to try to reduce the amount of, it's the only curative way to treat patients in AML, but also to try to reduce the amount of relapse. NK cells are the first lymphocytes to arise post-transplant, and we think they're very important in the hematopoietic transplant setting, but they're usually deficient in function for a good amount of time after transplant. So we looked at 100 days post-transplant, which is a time point where NK cells are still deficient in function. And what we wanted to evaluate is if these molecules could rescue some of those deficiencies. And so what we did is we evaluated, again, chromium release of HL60 tumor targets with NK cells alone, NK cells plus BiKE, which we see a difference in functionality, and that had been published before, and NK cells plus TriKE. And as you can see, NK cells plus TriKE gives you way more functionality and pretty much rescues almost completely functionality of NK cells in this post-transplant system than the BiKE or the NK cells alone, indicating that this could be used as a co-therapy in the post-transplant setting. We then evaluated, so I was just showing you tumor target killing. We then wanted to evaluate functionality on the NK cells themselves, and what we did is we evaluated degranulation by CD107A and intracellular interferon gamma and TNF-alpha on post-transplant patient samples treated with, again, HL60 tumor targets and the BiKE, 1633, or the TriKE, which is 16-15-33. And the BiKE is the lighter line, the TriKE is the heavier line. And as you can see, the TriKE vastly outperforms the BiKE in terms of degranulation, interferon gamma production, and TNF-alpha production. And below, I just have the pooled data showing that this is highly significant for degranulation, interferon gamma, and TNF-alpha. I guess I'm going to have to add 10 seconds for every slide. No, you went one too far. Okay. So that was against a tumor target cell line, but AML, primary AML, is much harder to kill than HL60 tumor targets. So we wanted to evaluate if these molecules could drive killing of primary AML. And the reason why primary AML is harder to kill is because of those inhibitory signals that I was talking about before. So we're really looking and seeing if we can bypass with these molecules. And if you compare with the previous slide, maybe I shouldn't do that. Yeah, okay. So now that's another 20 seconds to the presentation. As you can see, with HL60 tumor targets, you get more degranulation, interferon gamma, and TNF-alpha than when you look against primary AML. So we can actually see it in this assay. But the key thing is that the TriKE, which again is the heavy line, outperformed the BiKE, which is the light line, in terms of degranulation, interferon gamma production, or TNF-alpha production. And again, this data is pooled, and we use two different AML targets, shown by the fill line and the square. And the important fact about these two different AML targets is that they contain different levels of CD33. So we see slight differences based on the CD33, but they're not huge. But if the primary AML does not contain CD33, the molecules don't work. And that was published by Anders Björnik 2 or 3 years back with Bike. So next, we wanted to go to the myelodysplastic syndrome setting, which is also only cured by transplant. And although it's not as severe as AML, and the important thing about MDS is that you can usually encounter more NK cells, the NK cells that are present within these patients have a lower ability to lyse tumor targets than the ones from healthy donors. And again, this is a chromium release assay, and we're looking, so these are backwards, at a 20-to-1 effector ratio or a 10-to-1 effector ratio. Healthy donor patient NK cells vastly outperform MDS patient NK cells in terms of lysis. And again, this is data from DFAF in the group. So we wanted to evaluate if the TriKE could kind of counter that defect in functionality. And again, we compared NK cells plus HL60 targets alone, plus HL60 targets with the 1633 Bike, or plus HL60 targets with the 16-15-33 TriKE. And in terms of degranulation, interferon gamma production, and proliferation measured by Ki-67, the TriKE outperforms the Bike and no treatment in every single case. So one of the issues with MDS is that the myeloid-derived suppressor cells within the environment really drive down NK cell functionality. And that's reflected in when we take the NK cells out of the patient and do functional assays, we see a very strong decrease in functionality. So DFAF generated a co-culture assay in which she's co-culturing, and in black we have the controls, which is only NK cells alone, and in blue we have the NK cells plus myeloid-derived suppressor cells, MDSCs, with just IL-15, Bike, or TriKE. And the interesting thing about MDSCs is that they express CD33. So the idea is that during the co-culture assay, if we're targeting these MDSCs quickly, we're able to kill them. And the readout that you're looking at here is after 5 days of co-culture, DFAF took the NK cells out, and then she did a killing assay, a chromium release killing assay with HL60 tumor targets. And as you can see, compared to treatment with IL-15 alone, with only NK cells or NK cells plus MDSCs, you see this huge decrease in functionality. You see a similar decrease in functionality with the Bike, maybe it's a little bit smaller, but with the TriKE, you don't see a decrease in functionality, indicating that at least in this co-culture system, the TriKE molecule can induce a probably killing and bypass of the negative regulatory effect of the myeloid-derived suppressor cells. So we're going to do a change of pace and look at what the TriKE does in terms of killing kinetics with the NK cells. And this was a collaboration that DFAF generated between her, well, our groups, and Karolinska Björn Alfeldt's group. And they have a technology in which they can visualize NK cell killing at the single cell level. And what they use is these microchips over here that if you look at one single well of these microchips, they contain a 9x9 well axis or further division of the individual chip compartments. And within each of these, they can load one NK cell and 2+ tumor targets. And they can label the NK cell, and they can also label the tumor target, and they have a reactive dye in the media that shows you when a cell dies. It's super low. Yeah, I mean, it's super low. And as you can see, so this is the size of the compartment, right? So this is a natural killer cell. So it's a very small compartment. But the neat thing is that they can actually track movement of the NK cell and evaluate the movement and killing kinetics very precisely on a per microwell basis. And I'm going to switch out of laser pointer. And this is, again, NK cells are in blue, tumor targets are in green, and when a cell turns red, the cell dies. And this is TriKE-treated well, right? So, and the only bad thing about the system is that you can only compare to the conditions. But as you can see, the NK cell very quickly, it's moving around all over the place and very quickly kills 2 tumor targets. It'll take a little bit longer for the last tumor target. This is over 12 hours, though, I should say. But they can actually track exactly how much this NK cell is moving and measure the displacement. Again, you can only compare 2 conditions at the same time, so we only compared TriKE versus BIKE. Okay, I'm going to go. They're already dead. But so this is basically pictures at different time points of this happening over 12 hours. And the BIKE is on top, and the TriKE is on the bottom. And as you can see in this one, we have 4 cells, 4 tumor targets, and 1 NK cell. In this one, we have 3 tumor targets. And they can't do this super specifically. So you usually do 2 plus tumor targets, and it's usually between 2 and 5, and 1 NK cell per well. But what you can see very clearly here is that with the BIKE, you don't get as much tumor target killing as with the TriKE. And they can measure for each cell, they can measure the time to first kill. So the time where to engage the first target and kill it. And when you treat with TriKE, you see that the time to first kill is much quicker than when you treat with the BIKE. Like I said before, they can also measure the displacement. And this basically measures how much the NK cell moved around in the plate. And the TriKE-treated cells basically displace much more than the than the BIKE-treated cells. And each dot is 1 NK cell here. So basically, we think the IL-15 moiety within the TriKE is driving faster kinetics and more movement of the NK cell. And the final measure is serial killing. So this is a very important concept for NK cells, because you don't want an NK cell to just kill 1 target. You want it to kill multiple targets. So in comparison with, so the BIKE is in black and the TriKE is in gray, the BIKE-treated cells basically kill no cells more often. But the most important measure over here is that the TriKE-treated cells killed 2 or more cells much more readily than BIKE-treated cells. So we've just bought a new piece of equipment at IncuCyte soon in the lab, and we can see similar things in these assays. I'm just going to let this run, but basically, in this assay, we're looking at 16, 15, 19 TriKE, and we're looking at Raji targets, so B cell targets, which express CD19. So we can label the B cell targets, and we can add a dye, which basically turns the cells green if they're killed. So what you're looking for is yellow cells. So yellow cells are tumor targets that have been killed. And yeah, the movies are nowhere as pretty in this one, but the key thing is this technology allows us to measure kinetics of tumor killing. And in this assay, what we did is we treated with the 16, 15, 19 TriKE. We treated with rituximab, which is the standard of care, or we treated with nothing, NK cells alone, or NK cells plus IL-15. And we can track killing every 15 minutes in this assay, or we can open it up and track it every 2 hours. And what we see is a much quicker killing kinetics with the 16, 15, 19 versus rituximab, which is a very, very good ADCC-driving therapeutic antibody. So this assay gives us a little bit more liberty to test a lot more conditions at the same time and look at killing kinetics in that way. So having verified all this data in vitro, we wanted to go to a preclinical in vivo system. And we use NSG mice, which don't contain their own immune system in the lab. And what we do is we generate a syngeneic model in which we have human AML targets engrafted into the mice, and then we put in NK cells and whatever treatment we want. And that's depicted by here. So we usually, in this model, we radiate the mice day 0 to condition them, and this allows for better engraftment of the NK cells. And we don't know if this, we think it has to do with the monocytes remaining within the NSG mice, that limit the engraftment. So when you irradiate, you actually create room and they engraft better. After radiation, 4 hours later, we engraft 750,000 HL60 Luc AML targets. And again, these cells express a luciferase cassette, which allows us to track tumor progression in vivo without killing the mice. We wait 3 days, and we inject 1 million NK cells, and then we start treatment. And the BiKE and TriKE are small molecules, so we have to treat on a daily basis. So we treat 5 days a week with nothing, BiKE or TriKE, and I'm going to show another study later on that we're also treating with IL-15. And we evaluate bioluminescence imaging, so tumor load, after luciferin injection at day 7, day 14, and day 21. And so basically we can assess tumor progression in vivo. And then we do bleeds either the day of the BLI or the day afterwards to evaluate NK cells in the blood of these mice. And basically here we have the results. I should have made this a little bit bigger, but we're looking at day 14 and day 21, and we're looking at tumor alone, tumor plus NK and 1633 BiKE, and tumor plus NK and 1615-33 TriKE. And as you can see, at day 14, at an early time point, the BiKE and the TriKE look very similar. But when you look at day 21, we've already lost 2 mice in the tumor alone group. And we see a high level of tumor load in most of the mice that have the BiKE, but tumor load is much lower in the mice that were treated with the TriKE. Now, when we look at the peripheral blood at day 22, we see that basically, the tumor alone treated mice don't have NK cells, as they should not. The ones that were treated with NK cells plus BiKE have lost all of their NK cells. And this is due to the fact that NSG mice don't produce human IL-15. So there's no IL-15 for those NK cells to prevail. But we see that the IL-15 within the moiety in the 1615-33 TriKE is maintaining the NK cells in these mice, and it's probably driving some expansion. So we see a big difference in NK cells between these two groups. But this prompted us to ask whether or not the difference that we see in vivo has to do with NK cells numbers or with actual ADCC being driven by the BiKE or the TriKE. And this is a very complicated experiment, looking at a lot of time points. We're just going to focus right here. So we treated mice with IL-15, which is this group over here, and NK cells, and these mice have NK cells present there. But we also did a dose titration of the TriKE molecule. So 20 micrograms, 100 micrograms, and 200 micrograms of TriKE. And the important thing is between these 3 groups, and you can see a difference between them in terms of tumor control, they all contain similar numbers of NK cells. So the 20 microgram and 200 microgram groups contain very similar numbers of NK cells, but there was a difference in the tumor load, indicating that the molecule is driving ADCC, and it's driving it significantly better than IL-15 alone. So finally, Melissa talked a little bit about adaptive NK cells in her talk last week. Frank Saihaki, Jeff, and Fate are developing an adaptive NK cell product. And adaptive NK cells are basically, we don't want to call them memory NK cells, but they're NK cells that have been primed by a viral infection before, and they've changed their characteristics. And one of the important characteristics about adaptive NK cells is that they can drive ADCC for CD16 better. Obviously, this is interesting for our platform because we're driving functionality via CD16. So what we did is we took patient NK cells from a CMV positive donor and a CMV negative donor, and we put them in our in vivo system. And we looked very early on because we didn't want the lack of IL-15 within the system to be the driver of differences. So we looked at day 7. And as you can see, when you add tumor targets, so all of these have tumor targets, and the tumor alone is right here, plus NK cells from a CMV positive donor versus a CMV negative donor, you don't see a heck of a lot of tumor control early on. When you add the BiKE, you also don't see very much tumor control in the CMV positive versus the CMV negative. But when you add the TriKE, you see a big difference between tumor control and the CMV positive versus the CMV negative. And as you can see, this difference is statistically significant. So this indicates to us that with the TriKE addition, the adaptive NK cells function better in vivo. And it creates the potential to combining the FATE product versus the TriKE product that we're generating at some point in the future to do a combo therapy. So in summary, and I guess I went a little bit faster than usual, but I have a couple more slides. The 16-15-33 TriKE enhances NK cell activation and CD33 positive tumor killing. It drives maintenance and expansion of NK cells both in vitro and in vivo. It amplifies the NK cell killing kinetics. It drives tumor ADCC in vivo in a dose-dependent manner. And it can enhance adaptive NK cell functionality against tumors in vivo. So we filed a patent for the 16-15-33 TriKE. We're filing an IND, and that's almost at its last phase. We also filed an IRB. And the next step was to create a GMP-grade version of this molecule. And Dan Valera, one of our big collaborators in this project, has a space at the MCT facility that we have on the campus. And this facility basically regulates production of GMP-grade molecules. So he did, I think it was 12 batches to generate enough molecule for phase 1/2 clinical trial. And we validated all 12 batches to make sure that functionality was the same. He also checked for endotoxins. And they vialed enough drug to create a clinical trial. So hopefully, within a couple months, we are actually going to start a clinical trial in which we're going to treat patients that have CD33 positive malignancies with 3 blocks of TriKE treatment. And this is continuous infusion. And within each block, we have 24 hours of continuous infusion of the TriKE times 4, so 96 hours total. So we're going to treat them with 3 blocks. There's going to be rest periods in between the middle. And we're going to assess primarily the maximal tolerated dose and secondarily a disease response. And the enrollment criteria for this is going to be CD33 expression in high-risk MDS, relapse or refractory AML, or also systemic mastocytosis, which we didn't add here. And the other criteria is all the patients enrolled in this trial need to have at least 25 NK cells per microliter of blood. Sarah Cooley is going to be the PI of this trial, and we're hoping that, like I said, this is probably going to go in the clinic within a couple months. So what are the future directions of the molecules themselves? We started generating molecules now with nanobodies. So single-chain variable fragments are composed of a variable heavy chain and a variable light chain. And we find that decreases a little bit the functionality, because if you have 2 single-chain variable fragments within the same molecule, we think there can be some cross-linking between the variable chains from one single-chain variable fragment and the heavy chains of the other single-chain variable fragment. So we started generating molecules with nanobodies. Nanobodies are derived from camels, sharks, llamas, and basically they're versions of single-chain variable fragments which don't contain a light chain and cannot bind to a light chain. They're more stable and they can bind deeper grooves. And so our next generation molecules, instead of having an anti-CD16 single chain, is going to have a nanobody anti-CD16. And we've run some experiments and they're much more functional. They also restrict IL-15 function within the construct a lot less, so we get much better IL-15 signaling. So we think that's the future. We also want to validate these molecules in mice that have a more complete immune system. So we're using NSG, SGN3 mice engrafted with CD34 positive stem cells, which generate NK cells, T cells, and B cells to evaluate the molecules, and that's in the near future. Also with Craig Effield, we are starting to look at primary AML mouse models, and there's an experiment running right now, so hopefully we will know pretty soon how well this molecules measure up with primary AML. There's a huge number of TriKEs that we're testing in the lab. We have a 16, 15, 19 TriKE against B-cell malignancies, which I showed a little bit of data before. We have a 16, 15, 133 TriKE, which is supposed to target cancer stem cells. We have a 16, 15 EpCAM TriKE, which is supposed to target solid tumors of an epithelium nature. 16, 15, we call it SS1 anti-mesothelin, which we're looking for lung cancer. The CSPG4 with Jim, that we're looking for a variety of tumors. We have an IGF-1R TriKE that we're working with Dipali to look at breast cancer, and a 1615 ROR1 that we're looking at sarcoma. So as you can see, we're trying to target a broad variety of tumors with this. And we are starting to design molecules that include within the TriKE platform checkpoint blockade antibodies, well, single-chain variable fragments, and also engagers for other costimulatory signals within the NK cells to further prime NK cell functionality. So finally, I'd like to acknowledge all the people that were involved in this work. This is done with a very tight collaboration with Jeff Miller and Dan Valera. Jeff and my group are involved in the functional testing of these molecules and also all the experiments that are necessary to generate the preclinical data. Dan Valera has a group that basically can generate these molecules in a bacterial system. Within our group, we're also starting to use a mammalian system, which is a little bit better accepted by industry. Most of the in vitro work was carried out by Alex Lemvik, Sami Chu, and Behia Kodal. Ron McElmurray and Jacob Toller's lab was involved in the animal experiments. Deepak Sarhan within the Miller group was involved in the NDS experiments and the killing kinetic experiments with the Ormfeld lab in Karolinska. Again, I'd like to acknowledge the Valera lab and also Bruce Blazer for critical input in the project and all my sources of support. Thank you.
We'll take questions, Dr. Lagerstein.
So you mentioned the IL-15 part of the TriKE stimulates some expansion, you think. Mm-hmm. So as that happens, then you would dilute out do you know how much you're diluting out the amount of the TriKE that's bound to the NK cells? And have you thought about using gene transfer instead to make something like the TriKE on the surface of the NK cells?
Yeah, so
to prevent that problem.
There's definitely approaches looking at the CAR NK cells, right? To gene transfer so having NK cells that have specific receptors against CD19 or CD33. And Brandon and you guys are working on CRISPRs to induce better IL-15 signaling. In terms of, I think the valuable thing about the TriKE is that it's a completely off-the-shelf molecule, right? And in terms of diluting out the TriKE, the idea is that the TriKE dilutes itself very quickly just by running through the blood, right? So if we inject TriKE into a mouse, within 5 hours, it's going to be gone. So the idea is that we're generating a continuous supply of molecule within these people. And, the second we stop giving them the molecule, functionality goes away. So I don't think it would dilute it too much. The expansion will take several days, and hopefully we're going in with saturating doses of these molecules. Yeah.
Hi, so one of the things, so first of all, this is obviously very exciting for us for, new options for refractory AML and relapsed AML. And one of the things I always worry about is the difference between a patient who's relapsed by having 6% blasts in the bone marrow versus, 100,000 blasts in the peripheral blood. So tumor burden. And have you, especially with the mouse experiments that you showed, it looked like you were treating kind of before leukemia engraftment, right?
Yeah, so the HL60 system is super quick. They engraft very quickly. So if you look at the bone marrow of these mice, it's completely packed with blasts by day 14 and day 21. It's a
but
very
you
strong
start
system.
treatment on day 1?
We start treatment on day 3. So we give them 3 days. So the primary AML system is going to be much more interesting, right? Because we're engrafting for 5 to 6 weeks in that system. Then we're putting the NK cells in, and we're doing the treatment. So the tumor has colonized the bone marrow in that system. And by the time we're looking in, we think that we have very high percentage of AML blasts within the bone marrow. So it will be interesting to see if we get functionality there. The idea is not to, I think in my point of view, eventually, the idea is not to kill a full tumor load on patients, is to use it as a co-therapy with something else, that it's getting the majority of the tumor load, and then you come in with the TriKE molecule, and you get rid of the remnants that are there.
But for the purposes of the clinical trial, that Jeff said that one's coming, right? Like it's
it's coming. So the clinical trial, we have to start with something that is.
And you don't have a tumor burden restriction for those patients.
I don't think so. The restriction though is if the tumor burden is very high, they're not going to have NK cells, right? So that's the restriction. So we're hoping that restriction will probably balance things off, but.
I see. Thanks.
So I have a question about the last point that you were talking about including a checkpoint inhibition in your TriKE format. Is it known if you can fit, say, a fourth molecule into that construct? I can't make a good four-wheeler pun right now.
Yeah.
Or if you would have to sacrifice one of those 3 functionalities in order to include checkpoint blockade?
No, so we call them, they're hard to pronounce, TriKEs. It's really TriKE. So it's basically 3 single-chain variable fragments in IL-15. And we've done this for, we generated one that is anti-CD16, IL-15, anti-CD22, and CD19. And they work better, they purify worse. So in terms of production, it's a little bit tougher to get them. But the goal is eventually, and we tried one with anti-PD-1, and I think that was a mistake because I think we're actually cross-linking the PD-1 on the NK cells and driving more inhibition and functionality, but we are gearing up to do one against anti-PD-L1. So we're going to start with a TriKE format, which probably doesn't have IL-15, and then move into the TriKE format. And one of the neat things is the nanobody allows for better purification of these, so we think we can increase that platform further with nanobodies.
So a basic question: when you is this on? It is. Okay. Oh, there it is. Got it. So when you add the TriKE to NK cells in that nano assay, and you see increased motility, do you require the target to see increased motility?
No.
So it just increases on its own.
Well,
and
yeah. So I should say. Again, so one of the limiting things with that assay, the one from Karolinska that you guys saw the video, is we can only test 2 things at once.
Right.
So we've never done, I don't think we've ever done one that we specifically added just NK cells. Although I should talk to DFAV because sometimes you get wells that tumors don't go in. So we should check and see if the motility increases. I will say IL-15 in culture, there's a researcher in Texas called Emily Mays, was looking at what CD56 expression does in NK cells, and IL-15 drives CD56 expression long term. And what CD56 expression does, it basically drives almost this cephalopod on NK cells that seeks around signals when it gets increased. IL-15 drives CD56 expression, so we think by itself it will probably drive more movement. Okay. We don't have that specific experiment.
It'll be interesting.
Yeah.
And just one other question. So are there other CD16 expressing cells like monocytes?
Yes.
And,
so there's.
And do these things target monocytes?
Yeah. So there's 2 types of CD16, right? And our anti-CD16 actually targets both types. There's CD16A and CD16B. CD16B is expressed in neutrophils, but it doesn't signal, so it's just GPI anchored. CD16A is expressed on NK cells, trophoblasts, and macrophages. So it would be interesting to look at macrophages and evaluate if the TriKE is driving their function.
Do they respond to IL-15? Monocyte macrophages, or?
I think they can. I'm not quite sure, but yeah, I think so. I'll check into it.
I was curious about like how long your TriKE is staying in the body. You said it's clear
It's very
by
quick.
So like where are these TriKEs are going? Are they being cleared by the hepatic clearance or renal
Yeah,
clearance?
so basically they get clear for the urine very quickly. The TriKE is 60 kilodaltons, and we've done some kinetic assays, and when we look at the blood, you get a pretty strong decrease after 4 hours. We haven't really done it very carefully to do a full titration curve, but it's within 5 hours, it's gone, and it's clear for urine, and it's probably cleared for the blood too.
Then like instead of like having like IL-15, would you, let's say like if you have a pegylation linker, which can have a longer circulation of your TriKE, do you think it's gonna maybe benefit for your targeting?
So we've been thinking about generating more stable TriKEs, and another option is to put an FC, a non-immunogenic FC within the TriKE, and that would basically give it the same longevity in the body as a lot of these therapeutic antibodies. So there's a molecule it's called L-303, that it's basically a non-immunogenic FC portion bound to IL-15s with IL-15 receptor alpha, which is in the clinic right now, and that molecule remains in the body for about 5 days. Just to give you a difference, we're talking 5 hours versus 5 days. There's a difference in terms of the bioavailability of the molecules, right? The bigger you make them, the lower the bioavailability. So we think we get much better bioavailability with the TriKE. The other thing too is if you get a negative reaction from the TriKE, you just stop giving them the continuous infusion, and within hours, we hope that the patient will be restored to normal. Versus if you're given a therapeutic or a much more stable version, the negative reaction will keep going on. So there's plus and minuses of the molecule, but right now what we're going in the clinic with is this version of it.
Any other questions? Okay, thank you very much.
All right, thank you.